So we'll make a gentle start, and really I was just going to spend a minute or two to introduce everyone and the reason that we sort of set up the meeting. So first of all, thanks for everyone who's shown up and taken some time out of your evening to come and learn a little bit about metabolomics. Hopefully we can give you a few different examples of how metabolomics is used in sort of sports science research, and a bit of an overall introduction to it in general as a subject area from David. So we've got 4 talks, as you know. The first talk will be by David Wishart from University of Alberta, Canada. So we've got a big time difference. So we managed to accommodate people from all over the place, which has worked out really well. I won't say too much more. David can introduce himself and what he's going to talk about. Then Juha Hulmi from University of Jyväskylä. I think I got that just about right. Hopefully, Juha, who's gonna give us a talk. Then I will talk, and then Henning Wackerhage from Technische Universität of Munich, who's going to finish off. So talks from a really sort of broad range of topics, and hopefully they give you an introduction to metabolomics. And I suppose one of the reasons that I really wanted to do something like this is because I'm by no means an expert, and it was because of other people who were sort of very welcoming and have taught me more about it, that's allowed me to get into the field. And I think often with those technologies that we're not familiar with, that's the first hurdle, is sort of feeling comfortable to reach out, and you have a sort of first step in understanding, what it is and how you might be able to use it. So that's kind of why we wanted to do it, is to give you guys that opportunity as well, and hopefully signpost you to some different labs and some different people who you could make contact with if it's something that you want to do yourself. And if not, we just hope you really enjoy it and learn something new as well. Okay. So I'll pass over to David. If we just give a couple of minutes, obviously, to allow getting the slides on the screen and everything, that would be excellent. Whilst that's happening, I'll just go through a few sort of housekeeping rules as well, if that's okay. With regards to questions, if you could type them in the chat, that would be awesome. And then at the end of each talk, we can just pick through some of them and hopefully answer them for you. If you could please just stay muted, we'll just keep the speaker as the only person who has their microphone on. That would also be really good. In terms of data that might be unpublished and so on, the speaker will obviously let you know whether that's sort of confidential and you shouldn't screenshot it. But otherwise, I think we were in agreement that you're welcome to take screenshots as long as the speaker doesn't tell you otherwise. Okay. And finally, I should also just let everyone know that the meeting is being recorded. There is no intention to share it at the moment, because we know we do have some unpublished data from some of the speakers. So, but it's just to let you guys know that it is being recorded. Okay. So I'm going to mute myself and hand over to David. And yeah, I just hope everyone really enjoys it and takes a lot from it. Thanks, David.
Thanks very much, Dan, and welcome everyone. I really appreciate the effort that Dan and his team has gone into putting this together, and also for being so accommodating for the time differences. As Dan said, my name's David Wishart. I'm a professor at the University of Alberta in Canada. So we're about anywhere between 7 and 9 hours difference in time with you right now. And I've been doing metabolomics for about 20 odd years, actually before it even had a name. I currently run a center, a core facility called the Metabolomics Innovation Center. And that is a core facility that helps support doing metabolomics, helping with the data analysis, performing the mass spec and NMR. And I'll explain a little bit more about that. As Dan said, this presentation is sort of to kick things off, to give some of you an introduction to metabolomics, but also to tell you somewhat, I guess, an opinionated version of what you should do and what you shouldn't do in metabolomics. And that's from trying this field and working in this field for more than 20 years. So I'll dive right into things right now. The structure is that I'll give you a brief introduction about what metabolomics is. You'll certainly hear much more about the applications of metabolomics to sports and exercise science. I'll talk about, I think, 5 or 6 areas that I think are really important. One is in the area of experimental design for metabolomic study. I'll talk about the types of samples and how samples need to be handled in metabolomics. I'll talk very briefly about the analytical platforms that we use in metabolomics. I'll discuss the differences between what we call targeted or quantitative metabolomics and untargeted metabolomics. And then the concept of whether you should do metabolomics yourself, DIY, do it yourself, or using core facilities. And then I'll briefly talk about data analysis. This could easily cover several hours, but I'll just talk about some of the issues I've often seen, and then I'll wrap up. So in terms of, and maybe I'm just wondering if I put up the wrong slide set here. So just hang on, and I want to check here if this slide wasn't supposed to be here. So hang on in case I've chosen the wrong one. Let me just check if I've ended up using the correct set here. Sorry about that. I'm going to go back to sharing the screen. So can everyone see the slide there again, just to make sure I'm not sending you into
Yep, that's on.
slide oblivion? Okay. Okay, so this is the right one here. So when we talk about metabolomics, I often use this diagram to illustrate the connection between the genome, the proteome, and the metabolome. The genes code for proteins, and proteins are really there to manipulate the metabolome to help catalyze reactions. I've put a picture of a canary at the top of this pyramid of life, partly to indicate that the metabolites and the metabolome is sort of the canary of the genome. A single base change in the genome can have up to 1,000 or even a 10,000-fold effect in expression of metabolites. So just like canaries in a coal mine, which tell coal miners of low levels of methane or high levels of methane, the metabolome is a great reporter of what's going on in the body or in the cell or in a tissue. The genome, proteome, and metabolome are all connected in some way, but as you climb up the pyramid, there's a different degree in terms of physiological and environmental influence. What you eat or drink or breathe doesn't affect your genome. It barely affects your proteome, but it has a profound impact on your metabolome. So the environment influences the metabolome, the genome influences the metabolome, and also physiology influences the metabolome. And for people interested in sports and exercise science, this is important to remember. And it's also important to remember that different organs, different tissues can have very different metabolism. The brain metabolism is very different than liver metabolism, which is very different than muscle or gut metabolism. All of them profoundly different paths that are preferred. And that's detectable at the metabolome level. The brain, liver, muscle all have identical genomes, so they're almost indistinguishable. They obviously have different proteomes, but at the metabolome, they're very different. So that convergence of the genome, of the environment, and the physiology all happens at the metabolome level. And so this is why metabolomics is hot. It's the reason why people are using metabolite measurements to start understanding the connections between all these different spheres of biology, genome, environment, physiology. There's also an interesting difference at time scales. The genome is supposed to be quite stable. Hopefully it's not mutating as you drink your coffee or just digest your dinner tonight. it is intended to be stable. The proteome, if you've had a big meal, is just beginning to start to respond now. And so, insulin will be going up, ghrelin will be changing a little bit. But metabolically, what you ate or just ate or what you're digesting is changing your metabolome quite profoundly and rapidly, and will continue to change it for several hours onward. So the timescales are quite different between genomics, proteomics, and metabolomics. And the things that you're measuring can be quite different. And sometimes the timescale issue for metabolomics is very problematic. In other cases, it's actually really interesting because things that happen over minutes and hours, or even the seconds timescale, can be detected using metabolomics. Now in metabolomics, there are a number of routes of doing the technique. This is a general workflow. You can usually start with tissue samples. Muscle biopsies is sometimes used. If you're working with animal models, you can take other organs. If you're working with solid organs or tissue biopsies, you'll have to do an extraction. Otherwise, you can go straight into biofluids. You can get urine or plasma or serum or blood, even stool samples. So in those cases, you have extracts or biofluids that you can work with. In almost all cases, the preferred input to metabolomics instruments is a liquid. So from tissues to liquids or just straight from liquids. And from there, we can do a variety of chemical analytical techniques. We can use mass spectrometry, liquid chromatography mass spec, gas chromatography mass spec, NMR spectroscopy. And in fact, the techniques for analyzing chemicals have been around for 50, 60, 70 years. What's changed over the last 15 years has been the introduction of data analysis tools to look at large numbers of compounds at the same time. So everything in the top frame and even to the point of the chemical analysis has been around for a long time. But it's the data analysis, the data interpretation, which has really revolutionized or transformed analytical chemistry to become metabolomics. I'll talk a little bit about the data analysis, but I just want to emphasize that that is the real key to making metabolomics hum. Now, metabolomics has applications in environmental science. It has applications in food and agriculture, but also primarily in biomedical applications. And I think most of the people at today's meeting are here to learn a little about those. I'm just listing a few applications, areas that we've worked in for a long time. Other colleagues have. People are doing a lot of metabolomics and applying it to microbiome analysis, looking at organ transplant, understanding the immune system, obviously applications in disease screening and precision medicine, drug toxicity. Metabolomics really got its launch with the drug industry, and they were looking at drug toxicity profiling. It's very popular now in diet assessments. There are commercial groups and metabolomics companies that are doing this. Biomarker identification, health monitoring, newborn screening is an application of metabolomics that most people don't appreciate, but it's probably one of the most successful applications of metabolomics that's never really talked about. Millions of people have gone through newborn screening. Just about anyone who was born in the last 30 years has been screened with a metabolomic test. And then obviously applications in exercise and sports medicine, which is why people are here today. So I've given you a brief introduction. It's not enough, I think, if you're brand new to it, but for some of you, at least you're sort of familiar with the concept. I'm not going to talk about these areas of experimental design, sample types, analytical platforms. And so I'm going to dive into the weeds of metabolomics a little bit more. So, having been involved in metabolomics for 20 years, I've seen a lot of experiments. I've reviewed lots of papers, lots of grants, worked with many, many groups and labs, and seen all kinds of things, some of which are fantastic, some of which are kind of scary. So I wanted to highlight some of the things that I've learned over the last 20 years. So typically, the most common failing in metabolomics experiments or studies is in the experimental design. Lots of people just don't really think through their study sufficiently. And there are examples where people have done really great analytical work, really great data analysis, but they just didn't design the experiment properly, and so it can't be published or needs to be completely revised. Typically, people have or choose study sample sets that are too small. They're not able to get sufficient statistical significance. Often they're not able to do or don't do sufficient validation. They may not have a control group, as an example, or they may not have enough controls, or they haven't controlled for variables related to age or sex or diet. The other thing that we're seeing now, particularly over the last 5 years, is that many of the better metabolomics groups and studies involve not just metabolomics alone, but they'll combine genomics or transcriptomics or proteomics. And that trend is existing, and I think that's something that people need to be conscious of, because otherwise they risk not being really current or sufficiently scientifically sexy these days. I think in terms of the design do's, remember to try and design your experiments carefully, thoughtfully, spend some time in the library, spend some time reading related studies that have been published in the area that you're looking at. You have to make a decision often about whether you're going to be doing a cross-sectional study or a longitudinal study. Both can be done. Sometimes you also have to make a decision whether the study is purely a hypothesis driven study, or it can be a discovery study. Both are allowed, both are reasonable. Sometimes people will simply want to do a biomarker study and just simply say, here are the markers. But many people also want explanatory information, so they may be looking towards a mechanistic study. Mechanistic studies are intrinsically harder and often require much more data collection, much more data analysis. Again, a common flaw in metabolomic studies is that people have not done cases and controls, or they don't have sort of calibration point or reference points. Or if you're doing something that's before and after interventions, again, you need to think about those exactly and really don't mix these up too much. A general rule about metabolomics, it's just general, is that you need 30 to 40 cases and 30 to 40 controls. You can do it with less, particularly with certain rare conditions or where it's impossible to get that sample size. But it's also a case that more is always better. It's not unusual these days to see metabolomic studies involving thousands of individuals, just as the same with GWAS studies. A common mistake that people make is they don't track information about their subjects. They don't, whether it's animals or humans, they don't get information about the diet or drug or supplement use. Often there isn't enough information about age or sex. So these are variables that are really important to include whenever you're doing a study. And if you haven't controlled for those, they can often lead to misinterpretation of your results. Metabolomics involves collecting living samples. Samples are metabolically active, so you have to quickly collect samples and quickly store them, and those have to be stored properly, usually frozen. -80 is preferred. You should track and record sample handling and storage throughout. You should always try and collect enough material. We've had lots of issues where people just did not collect enough, and so they couldn't really either replicate the studies or validate the studies or do enough of the sample processing that they really needed. There are also examples of people choosing the wrong kind of sample type to look at the particular physiological question. So, don't try to study urine if you're trying to look at the lungs, and don't try and look at cerebral spinal fluid if you're looking at muscle physiology. So those are sort of intuitive things, but surprisingly, a lot of people don't think of that. So there's issues about design don'ts. You don't want to paint yourself into a corner, but also don't do metabolomics as an afterthought. And we've had lots of people approach us to do metabolomics when they said, well, I've done genomics, I've done proteomics, here's the dregs of my samples, can you just do some metabolomics? Or here's a sample that we just, we're going to throw out, but can you do some metabolomic studies with us? That's not going to be very useful because A, you don't know the handling history. Often the samples were collected improperly. Most of the metadata that you need isn't around, so it's going to be a useless study. I think there's also cases where people say, I have a couple samples and I think it would be really interesting to look at, or you have a tiny budget and you can only do a pilot study of 5 or 6 samples. That's really not enough to really do a publishable study. As I said, the choice of sample type is important in metabolomics. It's not like genomics, where you can choose just about anything and you can still get DNA. Proteomics is a similar issue. You want to choose the right sample or biofluid that reflects the physiological stress or impact that you're studying. I think the other thing that I mentioned before is include those external variables: age and sex, diet, drug use, supplements, other things, disease conditions that could influence the metabolome. If you don't include that, then again, you're often fishing in the wrong pond. So those are some experimental design issues. And they also lead to this point about the simple sample types and sample handling, which is really important in metabolomics. So particularly in exercise metabolomics, although it's less so in other areas, the question is, should I use biofluids or should I use muscle or tissue biopsies? Obviously, tissues collection is invasive. Biofluids generally non-invasive. Collections can be expensive if you're doing tissue. Handling tissues is also difficult. So as a general rule, if you can, try to work with biofluids. In terms of types of biofluids, often blood is the preferred or commonly looked at, but there's blood, there's plasma or serum. The reason why people look at blood is because it bathes all tissues, it bathes all organs. It's a sampling fluid that lets you understand what's going on at a holistic level. But people need to remember that when you freeze blood, it hemolyzes, and that completely messes up your samples metabolically. So don't use blood. You can use either serum or plasma. We generally prefer serum, but it's a flip of a coin. Now, in addition to other biofluids, people can also look at urine. And that's a widely studied sample set, but not generally well appreciated. Now, serum, as I say, it has an appeal, or blood has an appeal because it looks at global physiology. But the interesting thing that people forget is that urine actually concentrates a lot of the toxins or exogenous compounds that you can barely detect in blood, but are highly abundant in urine. So if you're looking at pathological effects or stress effects, often urine is a great biofluid to look at. And again, it just highlights the point that different biofluids for different purposes to monitor different organ systems. Now, it's also possible to look at other types of samples. Sweat is something that people are starting to look at more and more. It resembles dilute serum or plasma. You can also use it to understand a bit about the skin microbiome. It's relatively protein-free, which can be an issue when you're processing samples. Cerebral spinal fluid is essentially what I would call the urine of the brain. It is a way of probing brain metabolism. It's also relatively protein-free. Stool samples are getting very popular in metabolomics and microbiomics. It's used to analyze the gut microbiome, but you can also learn a lot about the diet. These samples are not so easily processed as CSF or blood or urine. Saliva is another one that people are starting to use more and more. It's a non-invasive way of getting things. It allows analysis of oral metabolism. People also use it to look at stress, cortisol levels, oral microbiome. But there's lots of protein in saliva, and that can be problematic. Now, the other point that many people forget is that whether it's saliva, sweat, serum, or even urine, the fluids are metabolically active. They have cells in them. Even urine, which is supposed to be, cell-free, it will have enzymes and it will have bacteria in it. Blood, of course, is very metabolically active. It's alive. And so if you let things sit out for even more than half an hour, you can be in trouble. So you generally have to either quench or freeze samples quickly. So you can either freeze them rapidly, liquid nitrogen or dry ice, or you can do an organic solvent extraction, which kills off the proteins in the cells. And that way the sample is metabolically inactive. Once you've done your quenching, things should stay frozen for as long as possible. So if you're storing them, keep them frozen. If you're shipping them, ship them frozen. And even if you're trying to extract tissues, generally people powder frozen tissues so they don't work with liquid tissues. They freeze them and pound them up in a pestle and mortar and then extract them. If you have got a sample that's been collected using standard methods for genomics or proteomics, they don't care how long something's been sitting out, often for hours, even days. So most of those samples that are collected with those formats are not suitable for metabolic studies. We get samples sent to us from all kinds of places. We've had samples sent to us in buckets. This is not a good thing. If you are collecting samples, like urine samples, or blood, even if it's a couple mils of blood or it's a liter of urine, break these things up into small aliquots, because then you can sample those small sample tubes over and over again. Because what happens when you thaw a large bucket, things start getting active again. Enzymes become active, metabolic processes become active. Even some cells can exist through the frozen state and they start doing their thing. So freeze thawing causes problems, and so the smaller the volume, the better. And the fact that you don't have to work with a large volume, but rather small volumes spread out, means that you can resample. We've had issues with dried samples. I mean, people do work with dried blood spots. Amino acids and acyl carnitines are stable in dried blood spots, but most everything else is not. The drying process takes too long. The hemolysis that occurs afterwards causes all kinds of redox damage to many metabolites and changes their true values from known values quite a bit. Lots of blood spots are stored at room temperature. That's also bad and leads to really strange values. So those are just some points about sample handling and issues that we've seen over the years and things to be careful of. Metabolomics uses a number of different analytical platforms. You can use NMR spectroscopy, high resolution mass spec, triple quad LCMS, inductively coupled plasma MS, GCMS. All of those techniques are used. We use all of these in our lab as an example. Each has their strengths, each has their weaknesses. NMR is used primarily to detect water-soluble metabolites, and you can detect and quantify anywhere between 40 and 120 compounds. ICP-MS is done for metals and metal ions. It's sort of the gold standard. GCMS has been around for a long, long time, and it allows you to measure organic compounds, organic acids, volatiles, alkanes, and depending on the technique, you can measure up to 250 compounds. High-resolution mass spec is probably the most popular format. It's best for hydrophobic compounds and lipids. And if you're doing quantitative identification targeted, people will get between 50 and maybe 200 compounds. If you're doing untargeted with high-resolution mass spec, you can get 5,000 features or tentative compound identifications. Triple quad mass spec is what's used in the clinic primarily. It's for newborn screening. It's done for lots of targeted metabolomics, and you can get between 50 to 500 compounds. These days, more than 80% of published metabolomics studies use mass spec, so it is the most popular platform. They have different levels of sensitivity. NMR is the least sensitive. High resolution mass spec is the most sensitive. You can get between dozens to thousands of compounds. The advantage of using NMR is that most of the things that you're measuring are known. The disadvantage of high resolution mass spec is that most of the things that you're seeing are unknowns. Roughly 10% of the signals you see in high res mass spec can be identified. The other 90% we can't identify. Of course, that's really exciting because if you can identify something new, then that's a high level publication. But it also makes it really difficult to interpret. So there are pluses and minuses with using different platforms. And my general recommendation is to use as many platforms as possible because they often tell you different stories and they help confirm what one platform has tentatively identified. So this is just some of the points about the different platforms: NMR, GCMS, LCMS, the time it takes to prepare samples, the time it takes to run samples, analysis time. Generally, with high resolution mass, the data analysis is the bottleneck. For NMR, it's generally the fastest method, the most automated method, but it's the least sensitive method. So you can't kind of have your cake and eat it too. Each of them has their advantages and disadvantages. NMR is very quantitative. Untargeted mass spec is not quantitative. GCMS can be both. So people can run them in different modes, but generally as a rule, high resolution mass spec is used primarily for untargeted metabolomics, whereas GCMS, NMR, triple quad MS is used more for targeted. So that kind of leads me into this issue of, what should I do? Targeted metabolomics or untargeted metabolomics? Targeted metabolomics is a technique that's been around for decades. We didn't really call it targeted metabolomics, but it's the method that's used in clinical chemistry. It's identifying a set number of compounds that are predefined. You measure their concentrations very precisely using triple quad and a mass spec or NMR. You have isotopically labeled standards. You do lots of validation and checking. There are actually kits to do targeted metabolomics. Untargeted metabolomics is derived mostly from the concepts of proteomics. You use high resolution mass spec, use lots of statistical methods to sort of figure out what's different between things. And then you identify of the most statistically significantly changed peaks, then you identify those compounds. So targeted, you know what you're looking at, untargeted, you don't. But untargeted typically gives you thousands of signals or features. Targeted gives you hundreds of compounds. So here's some of the advantages and disadvantages of them. So in terms of targeted, there's relatively limited coverage. So typically you're measuring between 100 to 500 pre-selected molecules. Typically, what you do targeted for is hypothesis testing or for biomarker discovery. In targeted, we'll do focus on quantitation. Targeted methods are very fast and they're very amenable to automation, and they're highly standardized. Now, in untargeted, you get huge numbers of features, potential metabolites, 5,000, 10,000 or more. There's this huge potential for discovery, for hypothesis generation, which is one of the reasons why it's so widely used. But typically, untargeted metabolomics doesn't quantify things. You can only do relative quantification at best. Most of the time, it's very hard to quantify. Pipelines for untargeted metabolomics are very slow and very manually intensive, except for a very small number of labs. And as a general rule, it's sort of a free-for-all. Everyone has a different method. So it's very hard to reproduce untargeted data from one lab to another. So untargeted metabolomics is far and away the most popular approach. And it's appealing because of the broad coverage it potentially offers. Now, the issue with untargeted metabolomics, more important people are becoming aware of, is that it's not as reproducible as people thought. There's a huge variation from lab to lab tests. The other thing that's emerged is now with advances in targeted metabolomics, the coverage that targeted metabolomics gets in terms of the compounds that are identified is often equal to or greater than what you get for untargeted methods. So that's an important thing to remember. The other point is that target metabolomics is anywhere from 10 to 100 times faster. It's obviously more reproducible. And these days it's actually getting cheaper. Another part to remember, especially if we're working in the world of human physiology and medicine clinics, target metabolomics is easily translated to the clinic. As yet, no untargeted assay has been translated and likely can't be translated to the clinic. And so if you're thinking about clinical applications, quantification is key to translation. So now I'm going to talk a little bit about the idea of how to do metabolomics, whether it's something you should do yourself or whether you should perhaps farm it out or whether you should work with colleagues. If most scientists like to do the experiments themselves, they like to do things in-house. This gives you much more control. It gives you the opportunity to train students and postdocs. People learn. If you don't have the facilities within your own lab, you can at least do it down the hall. So if you've got a colleague, then that's another preference that people have. And this is how most science is done. But having been involved in the world of omics, proteomics, genomics, transcriptomics, metabolomics for the last 25 years, and walking through many universities, there's lots of cases where you see either failed core facilities, obsolete facilities, where people have tried this, but it just didn't work, or they couldn't sustain it. And this is true with metabolomics. Metabolomics is an expensive thing to do. The instruments that I've shown you cost millions of dollars. The techniques require people with generally PhDs. The maintenance of the equipment adds up over time. And so I guess the point is that, everyone likes to fly, but not everyone needs to own a jet plane. And so it's for that reason why some of these national core facilities have emerged, both for genomics, for proteomics, and now metabolomics. Now, if you really want to do metabolomics on your own, if you have a mass spec or you have an NMR facility, you can do it. And I would really encourage people to do it using kits. This is how actually I started in mass spec-based metabolomics. I used to do primarily NMR, but we didn't know enough about mass spec, so we got some kits and we started tinkering around, and we used the Biocrates kits, and we learned a tremendous amount. Since then, we've also learned how to make our own kits, and we started making kits for GCMS, for NMR, and for LCMS. And now these kits, just like if you've done kits for molecular biology or other things, really easy to use. They have videos, explanations. They're relatively inexpensive, highly reproducible. They do targeted metabolomics. Now, if you're using a core facility and you say, okay, I don't have my own instruments, I don't want to get into a do-it-yourself, I don't have the time to learn everything, you can farm it out. And so these core facilities have, millions of dollars of equipment, or millions of pounds, or millions of euros worth of equipment. They have highly trained staff, they're full-time. They often have multiple platforms. They can handle all kinds of samples. They have very comprehensive assays. They can offer both targeted or untargeted metabolomics. They also can help you with your experimental design, your data collection, data analysis, and your interpretation. Often, they're much cheaper and they're much faster than trying to do it on your own. And so they're both academic or national core facilities, and they're also commercial providers like Metabolome or Nightingale or Biocrates. So I really would encourage that if you're brand new to metabolomics, this is probably the way to go, or at least the most efficient and the best way. If you've dabbled in metabolomics, I would really encourage you to try and use kits and to probably adopt more targeted methods. This is a slide I won't go into too much detail, but different facilities have different capabilities. Some have mass spec, some have NMR, some have GCMS, some can do fluxomics, some can do imaging, some can do metal analysis, some will have large chemical libraries, some will have limits in terms of whether they can handle at level 2 or level 3 biohazards. Some will have data analysis or database provision. Some will do qualitative, some will do quantitative or targeted. So these are facilities in Canada and Australia, US, England, and others. It's probably not totally up to date, but again, you can see a fair bit of variation among the different core facilities. Now with costs and how much you typically would have to pay if you went to a core facility or if you went to a commercial facility, it depends on where you are, it depends on which facilities you choose, which type of instruments, the number of compounds you're wanting to measure, the types of samples. So there's not a uniform price, but I have, and maybe some of you have also tried this, if you inquire at different core facilities or different labs or different commercial services, the price can range from $300 or 160 pounds, or I guess 240 euros, down to $30. So about a tenfold variation for essentially the same service. So, it's like buying a car and having to pay either 1/10 or 10 times the price for the same thing. So shop around, ask questions, get quotes. I think this is still very much a buyer beware market. There hasn't been the standardization that I would have hoped to see. I'll put in a little bit of a plug for the work we do. So as I said, I work for the Metabolomics Innovation Center in Canada. And this is an example of the prices that we have. So if you're trying to convert them to US dollars, multiply them by 0.8. If you're trying to convert them to euros, multiply them by 0.7. And if you're trying to convert them to pounds, multiply the numbers by 0.6. So this is a very heavily subsidized service. So these are unusually low cost services. But you can see that the approach has different GCMS, LCMS, NMR, ICP-MS, the volume requirements, the numbers of compounds that are detected, the numbers that can be tested or have been evaluated, and then the typical costs per sample. And so this is, I guess I'd say, at the low end of what you would typically see for most metabolomics prices. But I think the point that I wanted to make is that it's generally very affordable for most metabolomic studies. Last part I'm going to talk about is in data analysis. And this is something where I could talk about for hours, but I won't. It is one area where we spend a lot of time developing software and databases and tools like MetaboAnalyst and the Human Metabolome database, many other resources. These are critical to making metabolomics work, but also critical for interpreting metabolomics data. Just like with other omics data, we really encourage people to deposit their data in repositories like Metabolites, which is based in UK, Metabolomics Workbench is based in San Diego in the US. Both share their data, so regardless of where you deposit it, it becomes accessible. There's a lot of freely available data analysis servers like MetaboAnalyst and Galaxy Workflow for metabolomics. This really improves reproducibility because people can apply the same things, the same methods you described. They have at least workflows, code that you can reproduce. You can generate exactly the same analysis time after time, and other people can do that identically. So this makes your data fair, F-A-I-R, the FAIR principles. You can also take online courses on metabolomics data processing and statistics. I've been teaching a course for many years, and you can access it through YouTube at this site just called bioinformatics.ca. You spell it all out in words. I think to do data analysis, you need to become familiar with concepts of multivariate statistics. And many people, especially younger students, are learning R to help them with their data analysis. So this is a programming language. The other point that people tend to do is that they just sort of upload their Excel sheets and then run it through their pipeline and then say, here's my result. I'm from the old school, but I think you'll find that most people who do metabolomics do what I do, which is look at your data. Actually take some time to read it. Take some time to plot some simple plots. What statistics really does is it just sort of makes your intuition more official. If you can't see a trend with your eyes, it's not going to be there. You can do lots of fancy statistics to make the trend appear, but someone's going to show you that it's wrong. If you can't see a trend with your eyes, it's not going to be there. And that's, I think, the bottom line message I'd say. The other thing that people forget to do in omics studies, metabolomics, genomics, transcriptomics, is to validate your data. You find something in a simple small cohort, you need to repeat it, especially if you want to publish. And about 50% of the time when people try to validate their studies, it doesn't work. And that simply is an indication that they've either undersampled or been highly selective in their data selection. So validation is, again, a great way of making sure your findings are reproducible. Look at how other people do their metabolomics. See what's been published. Look at the more prominent labs or noted scientists in the field and see how they analyze their data and try and copy them. I mean, that's what we're supposed to do, and that's how you learn. If you can work with these core facilities, or if you use them, they can help you a lot with your data analysis in their bioinformatics. They often have bioinformatics teams that can help answer questions for you. They're happy to do so. They're sponsored and paid by the government to do that. I think as a rule, quantitative metabolomics data is a lot easier to interpret and process than qualitative data. If you use quantitative data, you can use a lot of the normal values that are published, that are known for different biofluids and tissues, and that helps a lot with your interpretation. And I think people need to go much beyond just simply identifying up and down regulated metabolites or just reporting the P values or significance. That's just statistical reporting. It's not biological reporting. What you should try to do is instead identify pathways, look at signaling or stress or disease pathways, identify processes. This is much more informative. You can also go and use your metabolomic data to help identify biomarkers or develop biomarker panels or biomarker risk scores. These are really useful for people. They're useful not only for you, but for the public, for clinicians, for other scientists. Linking your metabolomic data to physiology or phenotype using visualization tools like metabolite set enrichment analysis really helps with your interpretation, but also with your publications and other people interpreting your data. Using pathway diagrams, network diagrams, connectivity maps, trying to connect your metabolites to genes and or proteins really helps with data interpretation. And then when you're publishing your data, try to use, build a story, try to make biologically relevant connections or conclusions. A lot of papers I see just sort of end at the end of their statistics and just pile on a whole bunch of plots, and that's the end. That's not sufficient to be published. In terms of data don'ts, don't expect to identify a novel metabolite. I've been in metabolomics for 20 years. I haven't yet to identify a novel metabolite, and I've tried and tried and tried. Across the globe, only about 5 to 10 novel metabolites are identified per year. So this is not something, it's like, finding a meteorite. It's just really hard. I think in terms of people tend to use very sophisticated classification techniques before doing any clustering techniques. That's dangerous. So always perform clustering, visualization, PCA plots before doing PLSDA or other kinds of classification. If you're doing biomarkers, try and keep the number of biomarkers to less than 6 compounds. If you're developing biomarker panels, don't use things like neural networks or support vector machines. Use interpretable methods like decision trees or logistic regression or decision tree regression. I think people tend to overinterpret their data and people tend to overassert their findings. We all tend to do that, but try to be modest. And then I think the other question that people tend to forget to ask is, does this make sense? And I think that should be your overarching question when you're doing any kind of omics study. So I'm going to wrap up because I've been over time, but we've just recently published an article on metabolomics in exercise and sports. We've done a systematic review with a group in Iran. And I think it's a useful overview of what people have been doing, sort of highlights some of the trends in the field, some of the problems in the field, and provides some, I think, useful advice. I think to wrap up, key points I want to make is that a good metabolomics study begins with some careful experimental design. My supervisor used to say an hour in the library saves a month in the laboratory. So now it's maybe an hour on the internet can save you a month in the lab. So 10 hours on the internet looking through papers and reading can save you basically a year of wasted work. Remember to choose, to handle, and to store your biosamples carefully using some of the advice I've given. If you're doing metabolomics and you're starting off, I really recommend working with targeted quantitative methods. If you want to do it yourself, use kits, or if you need to do it outside, use these academic cores or commercial suppliers. When you're analyzing your data, use the free stuff. A lot of it's very, very good, very well maintained, and used by 50 to 60% of the community. So it's not as if they're going to frown on you for trying to use cheaper stuff. These things have been developed, millions of dollars have gone into their development, and they're supported by large teams of people. If you're trying to figure out how to analyze your data, just look at how those people analyze metabolomics data in high quality journals. It's okay to copy or mimic other people's methods. That's what science is about. That's how we learn. So, the rule I say, at least in any study, is, formulate things carefully, collaborate carefully, and validate your findings. So with that, I just want to thank some of my collaborators at the Metabolomics Innovation Center, Liang Li, Christoph Borchers, Bill, Michael, James, Dave, and Ian, and then our funders. And thank you for listening.
Thanks very much, David. That was really fantastic introduction for everyone, I think, and some really nice take-home messages there as well. I'm just going to ask if anyone has any questions, just pop them in the chat. We'll have a couple of minutes for that. And whilst we're waiting for any of those questions to come through, I guess I had a question actually, which was I'm seeing some more papers using the Raman spectroscopy method, and I wanted to just sort of ask what your thoughts are on that and where you see that maybe going in the future.
Yeah, it's an interesting one, and it's one that's been probably more popular in the UK, I think, than elsewhere. And there are certain fields and areas in metabolomics where there's sort of a distinct national preferences, either for the technique or for the technology. I think it's a really useful untargeted method, one that can be used for fairly rapid characterization. It's more a chemometric method. So it's really, you can't get quantitation, or at least very good quantitation. But and I think there's the possibility of looking at other types of, rather than liquids, you can obviously potentially look at solids. And so I think whether it's IR or Raman, those are, I guess, useful from the untargeted chemometric perspective. It's hard to interpret the data, as I say, in terms of often pathways or in perspective of normal concentrations or abnormal concentrations, things like that. It's a little more difficult with Raman.
Brilliant, thank you. Marie Phelan, who actually runs the NMR Centre up at the University of Liverpool and helped me a lot with my work, has asked a couple of questions, and one of them was related to that, but the other one was, do you have a preferred method for stool processing? The literature is wide and varied.
It is. We did write a review about this a couple years ago. I think it's called the Human Fecal Metabolome, and we talked about the different processing methods. There are issues both with storage and analysis. So some of the current recommendation is obviously freeze things very quickly, or store in ethanol, which seems to be a reasonably good way of storing samples. There's a huge variation in water content. And so one of the recommendations we made, and I think more people have been following, is this idea of working with wet weight, dry weight, and calibrating or normalizing to those masses, so that you can say whether things are up or down in a relative sense. There are some issues with freeze drying where you lose some of the short chain fatty acids, which everyone wants to measure. But again, you can still work with the wet sample as long as it was calibrated to what the true dry weight was. So we recommended basically splitting the sample and freeze drying one part and then working with the other to process with your conventional metabolomic methods. So some fairly detailed recommendations in that paper. And as I say, I think more people are using that, and also using the reference values that we published in that.
Great, thank you. Henning has asked a really interesting question, which is that exercise is often about metabolic flux. How tricky is fluxomics? Should we stay well clear at the beginning?
So fluxomics is difficult, but I think there's now a number of, good reviews and good methodological papers that can allow people to do that. There are also cores that can do fluxomics measurements. So our core, not my lab, but one of the other labs in TMIC, can do fluxomics and flux measurements. And so I think, unless you have lots of tools and skills locally, it is something you can outsource, and they can do that for you. But it is tough. Yeah, it's not an easy one. We've stayed away from it.
Great. Okay. So there's a couple more questions. So Rob Rust has asked, he said, thank you for an amazing talk. There's two main questions. One was that blood was preferred, or serum, over tissue for the analysis, tissue such as muscle. Could you elaborate a bit more on that? And the second one was enrichment analysis, that MetaboAnalyst is extremely sensitive to the metabolites you enter, even from the same data set. Could you elaborate on using MetaboAnalyst for the data analysis?
Sure. So with the first part of the question, the issue of tissues, the issue with tissues is that A, it's invasive, B, it's hard to get enough samples often, and enough of a sample to do robust metabolomic studies. If you have the capacity to do it, and if you are able to get the samples, and if cost is an objective, then I can see that muscle biopsies are just fine. I mean, people have done some amazing work in that. But if you're resource limited, if you have, if there's issues of ethics, if there's issues of sample collection, then blood is often a pretty good proxy. It picks up a lot of stuff. I mean, the muscle mass overall in the body represents, the largest mass of organs overall. So you're going to get a lot of sampling from the blood that way. But the blood technically also has, quote, contamination from other organs. And so it's not as specific as blood biopsy or muscle biopsy. In terms of metaboanalyst and the MSCA enrichment, I think this is part of a general flaw with the tool. And it has a lot to do with the database that it's using, which is the KEGG database primarily. And so we've been working for a long time to build out the database so that it's much broader. And so we've been building out this thing called the small molecule pathway database and PathBank database. So that still isn't fully integrated into Metabolomics. And there's still some fixes I think that we need to do. I mean, it's okay, but it's not perfect. And I think you've highlighted, a weakness in just that structure. And the weakness, as I say, has the real over reliance on the KEGG database.
Great. Okay, I'll ask one final one. Bilal, your second question maybe I think could be quite interesting from some of the stuff that Henning is going to present. So we'll leave that. But the first one, thanks for a wonderful presentation. What causes variations in untargeted findings, standardizations, for example, with a question mark?
Yeah, there's a bunch of things that lead to variations with untargeted. So there's, the instrument performance declines over time. So, as you run sample after sample after sample, things get clogged, things get dirty. There's matrix variations that occur. There's, they call it batch variations as well that happen. Again, this represents instrumental performance that just, as things clog up or wear down. There's, particularly the matrix effect. You can see, and we've worked with people where they said, we're seeing a consistent increase in this metabolite. And then they ask us to go back and do a targeted study. And when we do the targeted study, where we're quantifying with authentic standards and isotopes, instead of seeing an increase, we see a decrease. And that decrease is real. Or in other cases, when they think they see something significantly changed in an untargeted study, we try to repeat it with a targeted study, and that is no longer significant. So this represents essentially experimental noise, which then is overinterpreted as a statistically significant increase or statistically significant decrease. Now, if you do a lot of calibration and quality control in your untargeted studies, you can reduce that. You can't eliminate it. But I'm speaking from experience, and this is data from pretty good labs who are doing what they thought was as carefully controlled untargeted studies. So there is this issue which has not been fully resolved in the untargeted community. That the experimental and instrumental noise, matrix effects, batch effects, batch controlling effects, statistical corrections that people apply to things end up doing strange things to your data. Now, there are some protocols that some labs are doing that I think are much better, but it's still the Wild West. And even if someone tries to replicate what someone says they did, then it's still hard. So I'm kind of down on untargeted studies. partly because I've been burned so many times with untargeted studies. But I certainly understand their appeal.
Brilliant. Well, thanks so much. That's a fantastic insight there. I think that we can learn a lot from. I'll cut the questions there. I don't think there are actually any more there anyway, and just say on behalf of everyone, thank you very much for your time and a fantastic presentation and taking time to answer the questions as well. I'll signpost people to your facility, the website, and your details as well in a follow-up email at the end, so they can reach out and get to you if they need to. So we will now just move on to the sort of 3 shorter presentations that are, I guess, examples of how metabolomics is being used in exercise science research. So the first of the speakers is Juha Hulmi, who is from Jyväskylä University. So I'll just pass straight over to Juha, and then again, if you've got any questions, pop them in the chat during the presentation, and then we can get straight onto them after Juha's finished. Okay, thanks a lot again, David.
Okay, I hope you can see my slides.
Yep, very clear. Thanks, Juha.
Yeah, hello everybody. I work in the Faculty of Sport and Health Sciences at the University of Jyväskylä. Actually, I think I'm so sleepy that I will take the video off, so you don't have to look at my face. So greetings from snowy Finland. Thanks, Dan, for organizing this mini meeting. I hope there is not too much of those don'ts of metabolomics in my talk that David mentioned. So my talk is divided into 2 parts, in vivo studies in humans and in vitro studies in muscle cells. I start with human studies. So these are physique competitors. So we first compared young athletic physique athletes to age and BMI matched so-called normal people from general population using quantitative NMR spectroscopy with Finnish Nightingale platform. These physique athletes were more physically active and had higher lean mass and lower fat mass. We observed differences in the levels of 129 metabolites between the 2 groups, and most notably, there was more favorable cardiometabolic metabolite profile in physique athletes. As an example, lipid profiles and inflammation markers were at more favorable levels in these athletes. Now the next question is, can these markers be further improved by caloric restriction and fat loss? For success, athletes in many sports, they need to have low body fat. This is especially the case in physique sports, which are judged on aesthetic appearance. There is a paradox in which, at the same time, low energy availability has many negative effects on athletes, but on the other hand, caloric restriction has been reported to have many positive effects on health, also in non-obese individuals. However, it is not known what happens if already very fit individuals, like physique athletes, restrict their caloric intake. So we next studied what happens to the serum metabolome of physique athletes after fat loss achieved mainly through reduction of carbohydrate intake and increase in aerobic training while maintaining high level of resistance training and protein intake. To investigate what happens at the physiological level, including metabolomics, fasting serum samples were drawn from these athletes at baseline, at the end of the fat loss period, and at the end of the similar length weight regain period. as well as from non-dieting athletes. We observed that the low energy availability decreased total and visceral fat about 50 to 70%. After voluntary weight regain, these did recover back to baseline, along with many other basic physiological changes, including serum hormones regulating metabolism. There is some red line over there. I don't know how it came over there, but doesn't matter for me. Next, we looked at what happens to NMR metabolomics after low energy availability. We observed that cardiometabolic metabolome profile is further improved in these athletes without changes in non-dieting athletes. More specifically, we observed improved lipid profiles and decreased markers of inflammation after fat loss. These changes were restored after voluntary weight regain in these athletes. No changes were seen in non-dieting controls. Next, we studied these athletes using LCMS, and these results are unpublished. 191 unique previously identified metabolite features were altered after the diet. Metaboanalysts suggested a especially significant decrease in the enrichment of the biosynthesis of the unsaturated free fatty acids. Indeed, the most uniform decreases were among unsaturated free fatty acids, except EPA, as well as in short-chain saturated free fatty acids and endocannabinoids. The most uniform increases were among bile acid derivatives and very long free fatty acids. Similarly, as with NMR metabolome following the weight regain period, almost all of these potentially positive changes dissipated, and only minor changes were observed in controls who did not diet. Just before the COVID-19 hit in Finland, we were able to replicate the study collection. This time we had both males and females, and these NMR metabolomics results are still preliminary. We observed that most of the previous diet effects on markers of cardiometabolic health were replicated. In this study, we were able to examine the athletes also before and after carb loading to the competition. So these results are completely new ones. This 1 week of carb loading did not yet restore most of the observed fat loss induced changes, suggesting rather long-lasting effects of the low energy availability. However, carb loading had some effects in metabolome, as shown in the example figures on the right. Please look at the red bars over there, which depict the diet group, while green is the non-dieting group. The statistics here are just preliminary because the data is really new. Carb loading, for instance, restored diet-induced changes shown marked with asterisk in, for instance, serum glucose and lactate, as well as in ketone bodies and beta-hydroxybutyrate and acetone. The increased ketone bodies after fat loss probably reflect increased fat use for ATP production. Indeed, by indirect calorimeter, we observed increased relative fat use. So to conclude these first results, lean physique competitors, they have favorable cardiometabolic metabolite profile when compared to BMI and age-matched normal people. So-called normal people. 4 to 5 month competition period fat loss achieved mainly through reduction of carbohydrate intake had further positive effects on serum metabolome, even in those already fit athletic individuals. 1 week of carb loading did not restore most of these cardiometabolic effects, but 4 to 5 month weight regain did. Obviously, this kind of a diet is not sustainable, and it has many negative effects if continued for longer periods. But we think that in addition to investigating what happens to the physiology and health of the athletes, this design may allow investigating the paradox of both positive and negative effects of low calorie or carbohydrate availability. Okay. So now I show results from our experiments investigating low carbohydrate availability in vitro in contracting myotubes. So completely different type of a metabolomic study. We first stimulated C2C12 myotubes using electrical pulse stimulation, EPS, in both high and low glucose conditions. We also analyzed the effects of EPS on cell-free media to exclude the possible effects of EPS directly on media and to roughly estimate whether metabolite uptake or release occurs. Both cell and media samples were analyzed using quantitative NMR metabolomics at our university. We first noticed in simple PCA analysis that the principal component analysis, especially in the media, separates the groups based on glucose levels as well as EPS, as you can see here on the right. Please note that in this and later figures, high glucose is abbreviated HG and low glucose LG. The EPS on media without cells didn't have any effects on metabolome, and thus we think that our EPS results reflect the effects of myotube contractions. 24-hour myotube contractions almost completely depleted cells and media of glucose in low glucose conditions. Many changes in metabolism were observed, of which some are shown in this slide. On average, the responses in metabolome were augmented when myotubes were contracting in high glucose media. To roughly estimate metabolite release by comparing the cell media and cell-free medias, we observed that EPS seemed to increase the release of several metabolites, of course, obviously as well uptakes. But I show here some of the release findings. So the release by EPS seems to interact with the level of glucose in cell media. We observed that, for instance, lactate, alanine, and acetate are released as expected to media after EPS, but surprisingly also ketone isobutyrate. EPS also increased the release of branched chain fatty acids, probably due to increased branched chain amino acid catabolism. To conclude this second part, EPS stimulates metabolism and metabolite release. Only high glucose media maintains glucose in 24-hour EPS. Greater glucose availability augmented many but not all of the EPS responses. I want to thank all the researchers, research participants, and funding for these studies. And thanks for listening.
Thanks very much, Juha. Some really nice insights from two really different models there, which is really cool. One thing that I found really interesting was the decrease in endocannabinoids that you saw in the weight loss study, which is really interesting. I mean, I think there are some studies showing that increases in endocannabinoids stimulate lipogenesis. So do you think maybe we're seeing the opposite there, that as we bring those endocannabinoids down, that's sort of part of the mechanism by which it's allowing lipolysis to occur as opposed to lipogenesis? Or is it not really been that much of interest to you so far?
Well, I don't really know. That's a good point. They are also regulating hunger, so I don't know. I mean, these at least they are in hunger. Obviously, there are changes in leptin and ghrelin and so on. So it may be related to that as well. And there are also some health effects of those. So yeah. I don't know really, but yeah, they are having some lipolysis and going on and so on. But I don't know whether it's related to that or not.
Yeah, no, really interesting. That's really good. With your in vitro study as well. Obviously, the way I suppose the titles worded that elevated glucose has a sort of an augmenting effect. Could we also assume that basically the opposite is true, that actually low glucose media is stopping any of those sort of adaptive responses from happening in the cells as well, because I think we've seen something similar recently where actually by mistake one of the students forgot to do the media change before stimulating their cells and didn't really get much response. And then when we dug into it, we kind of found out, oh, that's literally because you didn't do this. And we looked at your paper and thought, oh, wow, okay. So really, if you're going to do these experiments, maybe the take home is that you need that high glucose media in order to see any changes.
Yeah, that's a good point. So obviously you can call it either low glucose decreasing dose or high glucose augmenting. So I don't know. I just decided to present this way.
Yeah.
But yeah, you're obviously right. But of course, there are some changes that low glucose is augmenting. We know in humans, for instance, also that some mitochondrial adaptations can be augmented with the low glucose. So energy stress and increased AMPK may be augmenting something. So yeah, brilliant. But here we saw mostly augmented responses in signaling and metabolism with a high glucose. So
really cool. So we got one question in the chat from Marie Phelan again. Have you observed any differences in the male and female responses to the low carb diet and then the regain?
Yeah, very good question. And I was actually thinking today about that, but unfortunately the sample size is not very big. We lost some at the regain period because of COVID, so it's really difficult to say. And so far, I don't have any statistics, so we haven't been able to look carefully those data, whether there is something that we can say with good confidence because of the sample size. So far, I just showed the pooled data, so I don't know. I don't have any idea, but let's see.
Cool. And I'll just pick one of the questions from Bilal that's come through so that we can stick to time. How can one say the effects were positive in healthy test individuals? One could say they were in normal physiological condition without intervention. Does that make sense?
Yeah, that's a good comment, obviously. So one thing is that it is known that caloric restriction seems to have positive effects also on the non-obese, already healthy individuals. So of course, we cannot say for sure if the already very good levels, if they are improving, whether that's really, if we can see changes in some hard outcomes as well. But at least these markers are going to better direction. So let's see. But yeah, that's quite interesting what happens in those caloric restriction studies in animals and in humans. So maybe those will explain some of the effects.
Brilliant. Okay. So in the interest of time, I'll say thanks, Juha. Really interesting. And if anyone's got any more questions, perhaps direct them straight to Juha, and we'll share some contact details in an email to all of you guys who have attended at the end anyway. Okay. So I'll move on to myself and try and stick to time. So please let me know the thumbs up. Henning, I can see you. Can you see my screen well? Great, thank you very much. Okay, so I'm going to try and keep it as on time as I can. My name's Dan Owens. I'm from Liverpool John Moores University. I think we've heard some really good talk so far. Where my talk hopefully is going to take you is a little bit more down the avenue of how metabolomics can be used in applied sport and exercise science. So in more of I suppose a real world example. So I suppose a background as to why we got interested in exploring metabolomics really comes, if you've done any work in sports science research, you'll know that ecological validity is often really difficult to achieve. As you can see demonstrated by the late Bengt Saltine here on the right, trying to get the correct sample at the right time in the most valid situation from the right sample population is really difficult to do. And so there's always been this idea that if we can get, minimally invasive, high throughput approaches, they could be attractive to try and get a more holistic understanding of athlete responses to training, competition, and nutrition, notwithstanding some of the points that David made in his talk earlier. So in the data set I'm going to talk you through today, we showed that metabolomics could reveal differences in marathon runners' metabolic responses to a marathon race when they were treated with probiotics or a placebo. So I'll just start with why we even got interested in probiotics in the first place. And this was really due to some of the work from Jamie Pugh, who's a researcher at Liverpool John Moores University as well, who showed in 2018 that 70% of the runners in the Liverpool and Dublin Marathon experience some gastrointestinal symptoms. And around 27% of those reported their symptoms to be moderate or worse than moderate. And those sort of symptoms typically manifest, as you can see on the slide here. So bloating, diarrhea, abdominal cramping, nausea, vomiting. And of course, if you think about what we're trying to do in a race situation, this is supposed to be peak performance. So these types of symptoms could clearly have a big detrimental effect on performance. And the reasons that sort of underlie these are thought to be oxidative stress, hypoxia, or redistribution of blood flow away from the gut, consequently a malabsorption of carbohydrate, mechanical stress of running, so that up and down mechanical movement can cause some gut damage, and hypothermia as well, or overheating the cells in the gut as well. And so there's been this sort of focus on solutions to try and mitigate some of these different symptoms and maintain performance. And one of those potential solutions has been probiotics, for reasons away from sports science research, from sort of the medical literature, which has shown the probiotics can enhance epithelial cell barrier function, increase tight junction integrity, so those junctions between the epithelial cells. They can have some sort of modulatory effect on the immune response and enhance communication between the immune system and the commensal microbiota, and also upregulate some antimicrobial peptides and antioxidant compounds as well. So there's been this sort of thinking that potentially probiotics might confer some benefits to endurance athletes and potentially mitigate some of the problems that we see with the gastrointestinal system during prolonged performance. So for our study, we organized a race, so a competitive race. There were prizes as well, and attracted some really good high class athletes to the event. We had a relatively small sample size, so we had 24 individuals take part. We had 20 males and 4 females, and they were randomized into 2 groups. So we had 10 males in both groups, 2 females in both groups, but the randomization was done based on their most recent marathon time. So the sort of performance level between the groups was matched. And they received a multi-strain probiotic or a placebo control for 4 weeks before the marathon race. One day before the race, we gave out a sort of a pre-race diet for the athletes to follow. So it was relatively high in carbohydrate at 8 grams per kilo, and pretty low in fiber. 2 grams per kilo of protein and 1 gram per kilo of fat. And we asked them to record everything they ate and drank via the 24-hour remote food photography method. So basically they would photograph everything they did, send it to us, and then we analyze that by dietary analysis software. On the day of the race, they arrived at the track, and before we gave them any food, we took a resting blood sample and a biopsy from a subgroup of all the athletes. We didn't force everyone to give us a bit of their beloved muscle tissue, but some athletes did. unfortunately, that's quite a small sample size as well. But nevertheless, everyone gave us a serum sample, so we had a full data set for that. So in the pre-race meal, we gave everyone the same thing relative to their own body weight. So they had a relatively high amount of carbohydrates, protein, and fat. So they got everything they would need for a best practice before a marathon race. They then undertook the marathon, and immediately post, we took some more blood samples, and if they volunteered, we took some biopsies again. And thankfully, right near the track where this took place, we had the ability to process the samples straight away and get them in a -80 freezer. So when it came time to do so, we prepared the samples for NMR. So unlike the other two talks that you'll hear today, we actually used NMR. Following that, we processed the spectra, assigned the peaks using Genomics, and then we deposited our data in a public repository, so that's available. And then, as you've seen earlier on, we performed the type of statistics that you will have, that you heard David talk about as well. So first of all, we normalize and scale our data using a probabilistic quotient normalization and Pareto scaling. And then we just did some univariate and multivariate analysis before trying to figure out where some of these metabolites might fit in terms of pathways. So just to give you an indication of how that sort of plays out and how it looked, it was a real big team effort that required us to be able to really get organized and get the samples from the athletes into the lab, processed and in the freezer as quick as we really could. And I should mention that during the race, the athletes received sort of best practice carbohydrate fueling. So it was at 60 grams per hour in the form of all these gels here. So firstly, just to point out what the probiotic did in terms of actual gastrointestinal symptoms. So first of all, what we saw was that in the latter third of the race, in the bottom left-hand panel, we actually saw a significant reduction in global GI symptoms in the probiotic group. So it seemed to have some positive effect on reducing gastrointestinal symptoms, but also allowed the athletes in the probiotic group to maintain their marathon running speed in the last third of the race, which was also really interesting. Now, when it came to the actual metabolomics analysis, first of all, you'll probably notice that the analysis that we did on the skeletal muscle was pretty poor. We couldn't really find anything out from that, likely because of everything you've heard earlier from David, which is that the sample size for those was really small. But what we did think was pretty interesting was that in our placebo cohort, we saw a bigger separation pre to post marathon in their metabolome than we saw in our probiotic group that was a little less disturbed by the insult of the marathon. On the right-hand panel, you can just see all the metabolites that changed across all groups and how they differed between groups. And this is represented as a fold change. And mostly what you're seeing is what has been reported before, actually, from the marathon metabolomes that we see. A lot of amino acids, we see some metabolites related to ketogenesis and also related to carbohydrate metabolism. So we took this forward and created a partial least squares discriminant analysis model to try and figure out which metabolites were the most important for the separation between groups at post-marathon. And you have all of this list of metabolites along the right, which were the most important. So these variables of importance and projection for this model that you see on the left. And again, we're seeing a lot of amino acids, particularly branched chain amino acids. We're seeing carbohydrate related metabolites, carbohydrate metabolism metabolites, but also again, some metabolites signifying ketogenesis as well. And one that popped up earlier on and also here is this metabolite desaminotyrosine, which we think is also quite important, and we'll follow it with some validation work. And I'll try and explain why in the next couple of slides. So we got all that information and we say, okay, well, we've got all these metabolites that seem to change, but does it really tell us anything? What can we learn from it? And so we tried to look across the other studies that we've done and see if there's anything that's telling there that we can sort of start to create a story and a picture. And so Jamie performed another study, which was more of a laboratory controlled study, in which, again, the participants received the same probiotic and placebo treatment for 4 weeks. This time they came into the lab and they performed 2 hours of exercise on the bike at 55% of the VO2 max. So typically the type of intensity that you would see on a long ride or the majority of a ride in a race, taking away the sort of the sprint or the main efforts. And what Jamie did was also use stable isotope traces here in order to understand whether exogenous glucose provided as a drink was better absorbed through the gut and utilized in the group that had the probiotic treatment. And first of all, in the placebo treatment, sorry. So what you can see in panel A is exogenous glucose in the probiotic group was increased above that of the placebo group with no change on that being produced by the liver. And then if you look at oxidation rates at the bottom, we're seeing a subsequent increase in carbohydrate oxidation. This is in the second hour of the trial, and a decrease in fat metabolism, fat oxidation. And then if you look at the labeled maltodextrin utilization specifically, we're seeing an increase in the utilization of that exogenously provided carbohydrate in that second hour of the exercise on the bike. So why do some of the metabolites that we see in the metabolomics analysis seem important for some of this or create a story? Well, desaminotyrosine has been shown to be a sort of a gut microbe derived metabolite that has effects on local and systemic inflammation, but also has protective effects over gut barrier integrity. So if we follow up from that and we look at some of the other effects that we see in our placebo group, we're seeing metabolites that are indicative of more of a reliance on the glucose alanine cycle to provide energy for the muscle to keep contracting. So as glycogen diminishes later into the marathon, the placebo group are relying more on the glucose alanine cycle to try and provide some energy to maintain those rates of glucose metabolism. But we're also seeing a concomitant increase in ketones, so 3-hydroxybutyrate. We saw increases in acetoacetate and acetate as well. So this kind of led us to the hypothesis going forward that if we can provide sort of best practice carbohydrates at, let's say, 60 to 90 grams per hour during endurance exercise, perhaps probiotics can increase the microbiome diversity that confers beneficial effects on the gut, allowing maintained carbohydrate absorption, and therefore maintain delivery of carbohydrate to the muscle to provide a fuel as glycogen stores deplete. So potentially for events lasting 60 to 90 minutes or longer, this could result in maintained carbohydrate oxidation. It might reduce gastrointestinal symptoms because we don't have it all sitting in the gut, and it's able to be effectively transported into circulation and possibly maintain race performance. Now, of course, we need to do validation studies following up on this, but I think the whole picture together is starting to create something that's quite promising and allow us to focus more directly on some metabolites that might be of interest. So the caveats to this work, and I think this goes for any of these sort of exercise related studies, is that we pick out these metabolites and say yes, they could fit in here, and other studies have shown that they're part of this pathway and so on and so forth, but we didn't and couldn't verify where any of those metabolites are actually coming from or where they're going to. So this comes back to the sort of flux question that Henning had earlier. Secondly, we couldn't verify flux through any metabolic pathways. And also our data might not translate right through to the very elite. So whilst we did have national standard athletes taking part, some of them are national standard, these data might not translate to the real elite. And because of the sample size that we have to be cautious about what the multivariate statistics in particular are showing us. And finally, just to carry on from one of the points I made there, when we do this sort of work, I think this paper that was published recently really nicely highlights what we should bear in mind when we're trying to translate those findings into practice. So yes, whilst there's probably very little risk of probiotics having any detrimental effect to sort of elite athletes, whether it's going to have such an effect that we saw in our cohort in these tier, particularly 4 and 5 athletes, we really need to be careful about making those claims. It's much easier for us to get tier 1 and 2 athletes into the lab and into our studies, and we can have much more powerful data and do better statistics on it. But do they represent tier 4 and 5 athletes? That's what we always have to bear in mind. So just to finish off, I just also wanted to signpost you to some of the other studies that we've done and we're currently doing. If you're interested in how we've used NMR metabolomics in elite rugby union match play, please read this paper. This is also open access in the same paper, or I should say issue as our probiotics study. So both of those papers are in there together. That was done by James Hudson, who is a PhD student at John Moores, but also at Gloucester Rugby. He's the nutritionist there. Sam Bennett, who's a PhD student with us, did some work with French triathlon and at the French Institute of Sport. We've got some really nice data on that, particularly around carbohydrate manipulation and heat stress. And then we're also trying to look at training and match responses. We've done a little bit with Liverpool Academy and Alberta, I think, who is on the call today as well. We're also going to do some work around match play in elite youth footballers with Juventus as well. And finally, just to say thank you very much to the guys who made the study happen that I've presented here today, particularly Marie Phelan, who I know is on the call, who really, introduced us to metabolomics and NMR. She runs the core facility up there and has been really fantastic the whole way through, and the support that we've received has been incredible, really. Andy Sparks, who let us use the track up at Edge Hill, and Jamie at LJMU, who this is a lot of this was his idea. And my sort of advice on all this is that, yeah, definitely collaborate and get people who really know what they're doing to help you out, particularly in metabolomics. Use those core facilities, as David mentioned, because it makes the world a difference. And yeah, thank you for being here this evening and listening to my talk as well. You can contact me if you want and ask any more questions, but I'll leave it there, and hopefully I haven't talked for too long. Nope, I think we're all right. Okay, so I'll have a little, if anyone's got any questions, that'd be good. If not, happy to sort of stay on at the end and answer any if we want to get onto Henning. Let's have a look. Okay, very interesting. Is there some evidence to assume that symbiotics plus probiotics should be preferred over probiotics in isolation? Well, potentially, the area now, we've even got postbiotics as well. And there's a lot of emerging evidence that different formulations, whether you have pre or post, or pre alone or post alone, have more beneficial effects than just probiotics on their own. And I think it's definitely an interesting question and probably where the field's going to go, but there's still so much to be done, even looking at probiotics, that I couldn't say that it's preferred over probiotics alone at the moment. And I also really can't say whether it's preferred just over fermented foods. One thing that fascinates me is whether we can see similar beneficial effects from food. I work with a lot of athletes, and one thing I always try and do is get them to avoid, where possible, having too much reliance on supplements, and really taking a food first approach, not food only, but food first. And if we could really get similar beneficial effects from increasing fermented foods in their diet, that would be amazing. So I'm really keen to do that sort of research as well and figure out whether we can do it naturally as well. But yeah, it's a great question. It's a bit of a minefield at the moment. Cool. Okay, so we're doing well for time, actually, pretty much bang on. So I'll any other questions, please email me, drop me a message on Twitter. Be happy to get back to you. And I'll hand over to Henning just to finish us off.
Yeah, thanks a lot, Dan. I hope you can see the slides.
Yeah, all clear.
Okay, super. Yeah, so I mean, it's fantastic also to listen to David Wishart and all you guys. I've just reread David Wishart's review in Physiological Reports, and I can really, really recommend it because it's a fantastic resource for everyone who wants to do metabolomics. So I present some of the work that our team here in Munich has done, and I'm just one of many who's involved in this. And we've started when I came from Scotland in 2016. So we got into it, developed some ideas. We put a team together, and what Dan has said is and what David has said is it really helps to collaborate in this work rather than trying to buy a fancy machine and then hope for the best that you get some data out of it that make any sense. So my title is Exercise Metabolomics and the Analysis of Physiological Extremes of Human Metabolism. And this is a meta-extreme study. And our question was, do highly anabolic, glycolytic, or oxidative athletes have unique blood metabolomes? So just to set the scene, well, the blood metabolome is influenced by many factors. And so one important factor, and Gabi Kastenmüller here in Munich, she was part of a big study published in Nature Genetics in 2014, and they did a genome-wide association analysis between the genetics and blood metabolite concentration. And actually, sort of your blood metabolites, you really have a unique fingerprint of blood metabolites, light concentrations in your blood. So that is quite unique, and it depends to quite some extent on genetics more than many other things. Of course, as Dan has mentioned, the microorganisms play an important role because, first of all, your own metabolism will sort of select microorganisms in your gut and other places in the body. But at the same time, microorganisms then secrete metabolites and add to your metabolomes in various tissue. Then some metabolites leave your body by excretions through urine and feces, and you put them back in. And here I've done a Munich student diet, so some beers and chips and stuff like that, but Dan would probably get a heart attack seeing this in an athlete. And but also it's not only nutrients, but also xenometabolites. For example, if you're a smoker, you put a lot of nasty stuff in your body. With exercise, and the different types of exercise, and what you do is with exercise, you don't add things to the body, but actually you activate certain bits of your metabolism, and that will change metabolic fluxes, and in the different compartments, also metabolite concentrations. And as an exercise physiologist, this is, of course, a bit we are interested in. So the specific background to the meta-extreme study was that we realized you can have a normal metabolism. So hopefully all of us, we are metabolically healthy. So we have a normal metabolism. And this is associated with a normal range of metabolite concentrations in the blood. But actually, if you then have defects of metabolism, and this can be acquired because you have the diet I've been showing with that big jug of Munich beer, lots of animal food and chips and all that sort of nasty stuff in the diet, then over time, this might turn you into a diabetic person. If you're aging, you're catabolic, or you have problems with your fat metabolism. And so these changes in metabolism, that sort of metabolic enzymes or metabolic transporters become defect, that you accumulate too much of metabolites, for example, given in your adipose tissue. Over time, this actually causes that your blood is changed and that you leave the normal range in terms of some metabolites. For example, given the best example is diabetes. So when your carbohydrate metabolism doesn't function, then if your fasting glucose exceeds 7 millimoles per liter, then you're a diabetic. Or if your LDL cholesterol is above 160 milligrams per deciliter, that's another issue. Or phenylalanine is above 120 to 130 micromoles per liter, then again, you're diagnosed with a metabolic disease. So on the pathological end, these changes in metabolism, when the metabolism is defective, then you get changed metabolite concentrations in the blood, so the metabolome will change. So what we now ask in our research is, so what is actually happening at the physiological end of the scale? Because, I mean, we are interested in athletes, and athletes actually have an extreme anaerobic metabolism. I mean, this is 400-meter runners, for example, given, and we actually had the double German champion in 400-meter running as a master student in our team. Then bodybuilders have an extreme anabolic metabolism, and marathon runners have an extremely aerobic metabolism. And so we asked the question, so if you have at the pathological end, at the pathological extreme of metabolism, you have changed blood metabolite concentrations, how does it actually look at the physiological end? So if you have extreme anaerobic, anabolic, and aerobic metabolism. And so we set out to do this. And this is Dani Schranner. It's actually her PhD project, and she's finishing her PhD. And so we wanted to compare sedentary controls. We wanted to sprint and power athletes, which are extremely glycolytic, endurance athletes, extremely oxidative, and natural bodybuilders, which are extremely anabolic. The limitation of our first study is the small sample size. It's a clear don't on David's list, but the strength is it's a big metabolic contrast, because of course, if you have a 400-meter sprinter, has a completely different metabolism than a natural bodybuilder, which is highly anabolic, or an endurance athlete, or a couch potato. And so the aims of the study were to compare the blood, specifically serum metabolome of controls, with that of these different athletes, which we essentially used as models for different types of metabolism. And then also we thought, well, okay, these guys, and that's well documented, have different enzyme activities in their muscles. So the sprinters have high glycolytic enzyme activities, these have a lot of mitochondria, and these have a lot of protein synthesis. But a lot of these enzymes and processes are only activated by exercise. So as a result of that, we wanted to compare them, the metabolome in the serum at fasted rest and after graded exercise test to exhaustion, which we again did fasted. We did all the experiments in the morning just to make sure to avoid circadian changes. Okay, so in the first experiment, and the groups have grown a lot, we had 4 sedentary controls, 5 sprinters, power athletes as a model for glycolytic metabolism, 6 endurance athletes as a model for aerobic metabolism, and 4 natural bodybuilders as a model for high anabolism. So these subjects were fasted, came in the lab in the morning, and more or less around the same time, Dani did an exercise test with them. And we took a serum sample at rest fasted, and after maximal exercise again fasted. And then on these samples, we did proteomics together with Marta Murgia and Matthias Mann. We did targeted metabolomics, which we've published, and with a much bigger cohort, we did then also untargeted metabolomics. We just got the data. We also looked at cfDNA. And we also put these sera onto some cells to see what is the effect on cells. Okay, so the next is now, let's have a look. If we look at the whole metabolome, and if you do a principal, something which is similar to principal component analysis, it's a partially squares analysis, so that we get an overall look at whether there are systematic difference in the concentrations of metabolites in the serum of these athletes. And so what we found is actually overall the metabolite concentrations in the serum of the controls, the sedentary controls, and the sprinters are quite similar. But we found that the endurance runners and the endurance athletes, they had a unique metabolite concentrations, and the bodybuilders, they also had unique metabolite concentrations, which really are away from the ones of the controls and of the sprinters. So it seems to be that, if you're a bodybuilder and if you're an endurance athlete, you essentially have different metabolite concentrations in your blood than the controls and then the sprinters. Okay, and then because I have little time, I just pick up one of these things where we found differences. So what we noted actually is that the bodybuilders, they actually had low amino acid concentration. So here's proline. On the x-axis, you see the different groups. So bodybuilding, control, endurance, sprint. On the y-axis, it's essentially a normalized metabolite concentration. The circles are the fasting rest, and the squares are the fasting post-exercise. And what you see is that the bodybuilders generally, if you looked at all the metabolites, they had the lowest concentrations. There were some changes with exercise. For example, given leucine, and most people seems to go down. But generally, if you look at that, the bodybuilders have the lowest concentration. And for some reasons, the sprinters quite often have quite high concentrations of these amino acids. And then we ask the question, so how can that be? The bodybuilders, they eat a lot of amino acids, or eat a lot of protein to promote their anabolism. And then actually we thought it's quite a simple explanation. So if you look at the controls, actually they have a relatively low muscle mass compared to the bodybuilders, and probably also a relatively low muscle protein synthesis. And you synthesize protein from amino acids. So if they are now fasted for a night and they have a relatively low rate of muscle protein synthesis, then they don't take up so many amino acids. And so as a result of that, they probably have a relatively high amino acid concentration when compared to the bodybuilders, which have a high muscle mass and a high muscle protein synthesis. And so if these guys fast overnight, then essentially their muscles are sucking the blood dry of amino acids. And that's our explanation. So we didn't do any fluxomics. That would be, of course, the experiment to do, or the classical tracer protein synthesis studies, but this is our explanation. Okay, since this published paper on the targeted metabolomics, we then also did some untargeted metabolomics. And now we had a much bigger cohort of 35 participants. And instead of looking at a limited number of metabolites, we now were able to quantify 850 metabolites in these 35 participants. And from all the 35 participants, we have 2 samples, again, fasted rest and fasted post-exercise. And interestingly, what we found is, again, we saw a similar pattern. So in this PLS-DA plot, which is similar to principal component analysis, again, the controls and the sprinters had relatively similar blood metabolomes, so similar overall blood metabolite concentrations. And again, the bodybuilders and the endurance athletes, they were similar. Another thing which struck us was that when you compare in the individuals control versus post-exercise, so we all know that if you exercise, the lactate concentration goes up a lot. But interestingly, if you then look at the overall blood metabolite concentration and ask the question, are they systematically different? And what is the effect of exercise on it? then actually the phenotype, so being a bodybuilder, being a marathon runner or triathlete, or being a sprinter, that has much more an effect on the overall blood metabolite concentrations than actually doing exercise. Yeah, so the bodybuilders, even when they did exercise, the overall blood metabolome concentration changes were relatively small because they stayed within the root circle here. So it was quite interesting that if you looked at 850 metabolites, that this was confirmed, that the results of the earlier studies were confirmed. Okay, and then this is now something which isn't metabolomics, but then we wanted also to know, so is there any activity in that serum? So when we exercise, does the serum actually change? And this change in metabolite concentrations, all that, what is the effect actually on cells? And here we use the model of human white adipocytes. And these adipocytes can produce UCP1, and then they generate heat, and that increases your energy expenditure. So it's just one example. And so we collaborated with the University of Bonn, and we took the fasted resting serum, put them on the adipocytes, and we took the fasted post-exercise serum, put them on these human adipocytes. So it's human serum on human adipocytes. And then these adipocytes, they can change their phenotype, and they can turn into heat generation. And so this is important for your health, because we know when this heat generation and the expression of UCP1 is turned on, that is actually associated with metabolic health. And so here are the data from this experiment. So what you see here is here are the controls, here are the bodybuilders, here are the runners, here are the endurance athletes, and here are the sprinters. And so we wanted to see whether this health effect, which is associated with UCP1 expression, whether we see that when we put the resting sera and the post-exercise sera on. Okay, so let's have a look at the controls. And so the circles are the resting sera put on the human adipocytes, and the squares are the post-exercise sera. And you see here in subject 1, in this control 1, you see that, yeah, if you put the resting serum on, the UCP1 expression is below 0.1. It was done in triplicates. And the exercise changes the serum. So if you then put the post-exercise serum of the same subject on these human adipocytes, then they switch on UCP1 expression. In subject 2, you see also a tiny effect. In subject 3, the effect is a little bit bigger. Subject 4 is a non-responder. Subject 5, subject 6, subject 7, whatever you do to the serum with exercise, but it seems to stimulate these cells to increase the expression of UCP1. So this is in untrained controls. And then we did the same experiment with the resting sera and post-exercise sera of the athletes. And here we were really stunned to see what happened. For example, given here in the bodybuilders, look at bodybuilder 8. Here is a resting serum. So we get on average probably a UCP1 expression, a relative expression of below 0.1. And then this absolutely shoots up when this guy was doing exercise, and we took the serum post-exercise. Something was added into the serum by some organ, and these factors now all of a sudden increased the RNA expression of this uncoupling protein 1. In subject 9, actually, already the resting serum increases UCP1 expression, and this increases further. But to our surprise, we also saw some subject 5 here, which couldn't do that. Yeah, he was doing the same exercise, and we didn't see this effect. And we saw similar things in the endurance athletes. Compare, for example, given subject 6, which is a complete non-responder, with subject 5, which really can switch on the heater, or more specifically UCP1 expression. Again, in the sprinters, we saw a similar phenomena. For example, given subject 2 is a complete non-responder, and subject 8 already, the resting serum increased UCP1 expression in these adipocytes, and the post-exercise serum had an effect. So this is now, so what is the point of this? So this shows actually when you exercise, you change your serum and you change metabolite concentration. And then if you put these serum on cells, the cells react to it. And so we, of course, now want to find out, is it the metabolite in here that is actually responsible for the stimulation, or is it other factors? So we do different omics methods to try to figure out which are the factors which for some people have in the serum that has an effect on these cells. And you can investigate the effect of exercise conditioned serum on cancer cells, on heart cells, on all sorts of other cells. And to combine that then with metabolomics and with proteomics gives you the possibility to maybe discover the factors which are causative. Okay, to wrap up, the question was, do highly anabolic glycolytic or oxidative athletes have unique blood metabolomes? And the answer is anabolic athletes, so the bodybuilders, and aerobic athletes, so the endurance athletes, have different blood metabolomes, so systematically different blood metabolite concentration than the anaerobic and the untrained subjects. The second point, when systematically changed metabolite concentration, whether a systematically changed metabolite concentration are just phenotype passengers. So that means, okay, they're changed, but the different metabolite concentrations in the blood of these athletes doesn't contribute to their performance, or whether there are some phenotype drivers in there. So that means that because a metabolite is changed, that helps the athlete to do a certain performance is unclear, and we really want to find out. Then what we also found is, and we think that is also a good study type, that you can do metabolomics, and then at the same time you look what is the effect of that serum on different cells like adipocytes, and then you can try to figure out, so which are the factors in the serum that then stimulate your adipose tissue or your heart or your brain cells. And so we want to do studies on that. And the applications of this research are that there might be blood biomarkers for hard to measure phenotypes associated with athlete status. For example, given we were hoping that we were seeing certain metabolites that had a different concentration in the sprinters, because the sprinters have a lot of type 2 fibers, and then that we maybe could validate that maybe metabolite A is a biomarker for a lot of type 2 fibers and for a fast twitch performance. Yeah, and this was one of the ideas of the study. And always have to thank all the people that have contributed to that, and always finish with a sporty photo near Munich. Okay, thank you very much.
Brilliant. Thanks a lot, Henning. That's a super nice talk, and yeah, some really interesting findings. There are two great questions, one from David and one from Mario. And I just had a thought as well, just before I got onto those questions, that one thing that came to mind was where you see those amino acid concentrations go down in the bodybuilders. This might support the idea, which has been contentious in the past, that pre-bed protein feeding might actually be really important for this group. I know the studies by Luc van Loon and so on were the caveat was always that the group who received the protein received more protein in general over the whole day, and so it was very difficult to weed out an effect. But actually, those data kind of suggest that pre-bed feeding could be really important for this type of athlete.
Yeah, I think so, because I mean, at the end, these guys have bigger muscle mass, or the percentage of muscle in the whole body is bigger. And presumably because of all their training, I mean, of course, I didn't train just before, but I assume that overall for the whole body, they have a greater protein synthesis. Mm-hmm. And protein synthesis needs amino acid as a precursor. And the more protein synthesis you have for a given volume of body, the more amino acids you need to take up from the blood. So as a result of that, you suck the blood empty. So as a result of that, the more protein synthesis you have, the more you suck the blood dry. And of course, these guys, they need to fill up their blood with amino acids. So they need to take in more protein. Yeah. So I think that is a practical implication of this research.
Yeah, brilliant. So David's question was, observation first into a question, was that the BCAAs for the sprinters seem very high. We often find levels of BCAAs to be indicative of a pre-diabetic state. How would you interpret the high BCAA levels in sprinters in terms of a non-pathological condition, or are the sprinters at a greater risk of developing diabetes?
Yeah, so we were aware of this diabetes issue and that there's an association, and in your review, you've discussed it also very well, David. Yeah, we're scratching our head, to be honest. So for example, given isoleucine here, I mean, it's clearly higher in this population with a highly glycolytic phenotype than in the oxidative, the anabolic, and the couch potatoes. And yeah, we don't understand this, why this is. Yeah, but I mean, they have more, surely, I mean, we had a double German champion 400 meters. So these were not just average athletes. So this was really a top athlete, yeah, who ran 400 meters in 45 seconds. And yeah, we wonder why these concentrations are high. So we can't explain. Yeah, so it's a really good question and a really good link to mention that there's an association with type 2 diabetes. Yeah.
Fascinating. I think definitely that, that's a clear example of where this will definitely lead on to another really interesting study to follow up now, because there's a really unexpected thing to pull out from it and clearly warrants further investigation for sure.
And the question is, we know there's a large variation in fiber type distribution. And so a big question is, of course, are people who have more type 2 fibers overall more prone to diabetes? So I mean, this is things you can follow up, because I mean, this is one of the biggest differences in human metabolism, because if you look at Claude Bouchard's studies, I mean, there's a huge variation in muscle fiber proportions. And muscle fiber proportion, type 2 fibers have a completely different metabolism than type 1 fibers. So, and it is also carbohydrate metabolism. And so is there some link that clever metabolite researchers can figure out, or metabolism researchers can figure out? Yeah, so this is also the attractive things of doing this kind of study, because you stumble upon things where you then start thinking, so hang on, how does it actually work?
Amazing, really good. And the second question was from Mario. Great talk, Henning. Congrats. Have you already checked factors / exokines could be responsible for browning? For example, cytokines such as FGF21 or metabolites such as BAIBA. I don't know what that metabolite is.
Yeah, to be honest, we did these experiments because there was a big consortium grant in Germany on this thermogenic adipose tissue. And this was actually a preliminary data experiment. And we got the grant, so we are working on the questions for the next 4 years. Yeah.
Awesome. And then there was one last question. We'll go with this one, and then we'll finish off from Bilal. Hi, Henning. Wonderful talk and findings. Did you investigate if the SERA UCP had influenced adipocyte metabolism? UCP induction post-exercise is well known.
So as I said, this was preliminary data for grant. We got the grant, and this is now, of course, for the next 4 years on the research agenda to try to identify the factors in the sera of the responders versus the sera of the non-responders. So one experiment we want to do, we want to put the sera of the responders that really switches on the heater, so the UCP1 expression on the adipocytes, and do phosphoproteomics. And the phosphoproteomics, because for example, given if SMAD signaling is affected, so it could be myostatin and things like that. Yeah, so if AKT is affected, and so essentially it is a little bit like playing Sherlock Holmes. You look at the evidence at the crime site, and then you hope you can deduce who the criminal was. Yeah, and so this is the strategy for which we got the funding, and this is what we're going to do in the next 4 years.
Amazing. Brilliant. So I think we've kept pretty much the time. We've taken an extra 10 minutes off people, but I'm sure you're all okay with that. At least I hope so. Don't send me any terrible emails afterwards. So I'll just finish with saying thank you to firstly the speakers for taking their time to do this. And this really just came about through a few emails back and forth, you know, and it's really great when people sort of offer their time and say, yeah, we'll help out together, what you can pull off. So thanks to the guys for getting involved, and thanks to David. I know, you know, you have such a busy schedule, so that's amazing. And thanks to everyone who's shown up as well. I know it's different times for everyone, wherever you are. It's probably quite late, particularly for Juha being a morning chronotype. I'm sure he's maybe asleep now behind the camera, but yeah, thanks to everyone. And look, if you have any questions, we'll follow up with an email that will go out to everyone who's attended, and we'll include contact details and signpost into the different labs and so on. So if you do want to get in touch, or you have ideas based on some of this really interesting data, you think people could collaborate, then that's what we'd really like to happen from this. So please do so. So yeah,
and thanks a lot, Dan, for organizing. Sorry for interrupting.
No
so
problem.
On behalf of everyone who was listening, and on behalf of the speakers, if I might do so, thanks a lot for organizing. It was fantastic that you put this together in 2 hours. It's a good time to get a good taste of that. So thank you very much.
Yeah, thanks a lot, Henning.
Yeah.
You're very welcome.
Well done, really well done. Thank you.
Thank you, David. Cheers. Thanks a lot, guys. So yeah,
Thanks,
enjoy the rest of your evening. Liverpool have just kicked off against Arsenal, so I'm going to be able to catch the football as well. So that's fantastic. But hopefully catch up with some of you soon, and maybe in the future we'll actually be able to have a drink or something at a conference when we see each other face to face. Thanks everyone.
Bye for now.
Bye bye.
Bye bye.